All right, let's start with a sound check. Can you guys in the back row hear me? Okay. Can you guys in the back row hear me? Excellent. Just as a refresher, and actually one small update, in terms of course matters, I do want to remind you and encourage you to take advantage of the review session that's going to be held Tuesday, it's tomorrow, in this room, 6 p.m. Susan's a very capable person and is a real resource, so I do hope you'll come. Some people have asked me if there are posted answers to the sample test that was given out or made available. Those answers are not posted. If you have an answer you have any doubts about, you're certainly welcome to come talk to Susan, to Angie, or to me, or to call Carissa and make a contact with her. In addition, so there's online is a practice exam if you want to get one down, or tutors you might want to work with. Some people had trouble finding the links to the Google version of the lecture videos. At the top of icon, there's a toolbar that has content and some other stuff and links. As you click on the links, that's where the links are, and they are. We had a few technical difficulties with Friday's lecture, but I think it should be available on the web in both versions now. This week, and this week only, Angie is going to reschedule her session to make it more useful for your study efforts and have it on Wednesday at 4:30 after this class, so you have a choice between my office hours or her, depending on what suits your style. Are there any other, well, I guess one other thing I could mention, a couple of people have asked. Whatever we cover today in class might also be on the exam. Nothing on Wednesday will be on the exam. So it's through today, and then we come to a stop, and whatever we talk about Wednesday, that could be on the following exam. Are there any other just kind of course-related questions people have? There are also some study questions down there if you didn't get them on Friday. Any other general course questions, questions about the exam coming up?
Are you being close to the answers to your class?
I did just say that I'm not going to. Or did you just come in? I think it's fairly generous to give out the tests, but to post sample answers is beyond my latitude. If you do have trouble feeling confident of an answer, do feel free to talk to Susan, to me, or to any of the other TAs. Any other questions? Okay, let's talk about real science then. Do people have any questions from the chapter on the cytoskeleton? People have anything they want to ask about what we talked about on the cytoskeleton? All right, well, if you have questions later, feel free to ask them. So now we're going to move into one of the premier areas of biochemistry, one of the things that has focused people's attention for over a century, one of the first places where there are real triumphs of getting glimpses into the chemistry of life, and that is talking about enzymes. Enzymes are, I want to make sure this is it's the first one. Enzymes are catalysts. And so of all the kinds of proteins that are made, there are proteins for structural purposes, there are proteins for recognition. Of all the different kinds of proteins that are made, the enzymes are those proteins that are capable of this wonderful feat of catalysis. And we're going to get at a glimpse at some of the principles of enzymes, or behind how they work, by looking at a particular enzyme named chymotrypsin. It makes a good model for a number of reasons. It's an enzyme that almost all of us have. It's a pancreatic enzyme, so it's made in the pancreas and dumped into the stomach as part of the digestion of foods to break down meats. It breaks down protein by hydrolysis. So if you look at a peptide backbone here, you can see that the peptide bond can have a water added across it, and now you have two fragments, one with a carboxylic acid at the end, one with a primary amine. It can do this 190 times a second. And so it's a real advantage over what would happen spontaneously. And to give you an idea of just how good it is at it, the fact that it is 10 to the 11th times better than without the enzyme is quite encouraging. Also suggests how stable proteins are in the absence of something going around breaking it up. If we look at it structurally, we see it has two domains, that there's a sort of a compact chunk here and a compact chunk there that are loosely connected by these folds. This cleft here in the middle is where the actual hydrolysis, the actual breaking of peptide bonds occurs. And we're going to look at that in a little bit more detail later on. Folks, I know with wintertime things get harder to move around. Let's try to make sure that the patch up there is kept clear so people who really can't get here on time can get in more easily. One of the advantages of chymotrypsin compared to other ways of breaking proteins down is that it can act under mild conditions. It can act close to neutrality, whereas if you were going to go out in the wild, you might need to get high pH or low pH to get a significant amount of hydrolysis happening. It has a measure of specificity. It's not excruciatingly specific, but it has some specificity, and we'll be talking about what makes an enzyme specific, what contributes to enzyme specificity. So that is the key of what an enzyme is that it makes a reaction go faster. You can speed up reactions by heating them, but most organisms live at a constant temperature, so that's not practical. Or you can do speed up a reaction with a catalyst. Enzymes are only one class of catalysts, of course. Things like platinum can catalyze reactions. The overall principle that defines a catalyst is it is not consumed in the reaction. And we're going to get to a place with RNA where someone calls something an enzyme, but it's not because it's used up in the reaction. But as catalysts, enzymes are really amazingly good catalysts. They exceed anything we can design as chemical engineers for most purposes. And if you want to get sort of a sense of just how true that is, here's ornithine monophosphate decarboxylase. So it's taking off CO2 basically. You wouldn't think that would be that hard. Their estimate for how fast it happens is it would happen, half of it would happen in 78 million years. And so the catalyzed reaction can happen 10 to the 17th more quickly than the uncatalyzed reaction. There are some other reactions here where the absolute acceleration is not that great, but the rate itself is remarkably fast. Because the uncatalyzed rate was somewhat quick to begin with. But this should give you some sense of just the extraordinary power to accelerate reactions that we see. This is only 2 million-fold accelerated, but the basal rate was 7 hours compared to 78 million years. So you can see that it's starting from a much higher baseline to begin with. Let me just make sure. Yeah. I did want to also point out that you can talk about how fast it is relative to the uncatalyzed reaction and how fast it is intrinsically. So this is showing the intrinsic rate. Okay. One thing to keep in mind about enzymes is that they often, not always, but they often have names that will tell you what the reaction is. And as we go through this course, I will be trying to point out when the enzyme name is not transparent, because it so often is that it's helpful to have it drawn to your attention when it's not. So for example, here we have pyruvate, 3-carbon molecule. We're going to see this molecule a lot, so get ready to get very familiar with it. The enzyme pyruvate decarboxylase removes a CO2. It decarboxylates it, leaving you with a 2-carbon fragment and a carbon dioxide molecule. Here's alanine, one of the amino acids that we're familiar with. And here's a dicarboxylic acid. There's a carboxylic acid here and a carboxylic acid there. This is alpha-ketoglutarate, another molecule we're going to get very friendly with. And the enzyme alanine aminotransferase takes alanine and transfers an amine. And now we end up with glutamate, because it has the amine now, and what was left behind is pyruvate. So the enzyme name is quite instructive in terms of what goes on. There are 6 classes of enzymes that are recognized. In the first class, they recognize oxidation and reduction reactions. The second is transferases, of just moving things from one place to another. Another is a hydrolase. We talked about chymotrypsin. That's a hydrolase. It breaks up something else. Lyases are ones that can eliminate a group and make a double bond at that spot. It's a little subtle of a reaction. Isomerases rearrange a molecule without changing its chemical nature. And ligases can create new bonds with energy that's been put into the reaction at the cost of ATP. And you can see that some of these things are very common. So there's bunches of hydrolases, which reflects the fact that we use them to feed ourselves. There are not so many ligases because it's an expensive reaction and you don't do it unless you need to do it. Oxidoreductases are pretty common as well, part of the energy chains, but also part of synthesis to make things in a more reduced, more complicated form. Enzyme kineticists came up a long time ago, or maybe chemists per se came up a long time ago, with a way of trying to represent what happens in a chemical reaction. So we can think about it in a little clearer way or try to represent what's going on. So if we think of a reaction here, and this is just a kind of idealistic platonic reaction, we have a molecule that consists of A and B and a third one called C. And if that mixture of stuff goes, reacts, proceeds into A, and now it's B attached to C instead of to A. Let's just call that our hypothetical reaction. That's our ideal platonic reaction. We can talk about the different stages of this reaction in a graph called a reaction coordinate. And so on the bottom axis doesn't have numbers so much as it has sequential stages in the reaction. And the vertical axis in this case is free energy, Gibbs free energy. So what we're looking at is the energetic state of the various components of the reaction. And for this particular reaction, well, in any case, for all of them, we can distinguish by looking at a graph like this what the free energy is of the reactants, the transition state, and the products. In this particular reaction, we have an initial state of reactants that has the same energy as the products. And the intermediate, the transition intermediate in the middle of the reaction is of a higher free energy. It's always the case that the intermediate has a higher energy. That's part of what makes it intermediate. By looking at the change in free energy, delta G. So delta G is the change in free energy. If we look at the change in free energy from the reactants at the starting point to the products at the end point, it will tell us whether the reaction is spontaneous. And by spontaneous, a chemist means you don't have to put energy into the reaction. It doesn't by itself tell you anything about how quickly. So the size of the energy gap won't by itself tell you what the rate is. Here we have some reactants, and they've gone through a reaction, and here's their product, and there's a negative delta G. This would be considered a spontaneous reaction. So the delta G here is negative. This is a spontaneous reaction. In this case, the reactants are somewhere else. Here's the products there. This has a positive delta G at the end of the reaction. It won't go spontaneously. If anything, it will go backwards. And we haven't yet tried to talk about the height of the hill in between, but that's going to turn out to be important. Everybody comfy with the reaction coordinates and what information we get out of them? Okay. What catalysts can do, and this is true whether it's an enzyme or whether it's platinum or whatever your catalyst does, what the catalysts can do is change the transition energy. They don't change the overall energy, just the transition energy. So in this case, we have some reactants, we have some products, there's a negative delta G there, so it's a spontaneous reaction, i.e. we don't need to put energy into it for it to go. In the absence of any catalyst, those reactants have to go through an intermediate that has this level of free energy. So there's a large free energy difference compared to the reactants. And then they can roll downhill from there to the products, because obviously that's negative delta G from there down, so it's going to fall down. If we have a catalyst around, the reactants are going to be the same. The products will be the same. The difference in free energy between the reactants and the products is the same. But what is different is the free energy difference between the reactants and the intermediate, the transition state. Or if you like, between the intermediate and the product. And so that is how catalysts can change the rate of a reaction without changing whether it's spontaneous, just by changing the transition state energy. And the reason this changes the rate is because it changes the amount of energy it takes to get over that hill. If you think of this as you've got a handful of marbles and you're throwing them up a little bump, if it's a big bump, not very many of those marbles will get over the bump. If someone comes and smooths the bump down so it's lower, and you throw marbles at it the same way, more of them will get over that bump and trip up your friend when they step on them, or whatever. So sort of imagine this with marbles kind of rolling up or downhill. Yeah, I'll just go back one more time. Any questions then about the reaction coordinate, what it's telling us, and this fundamental property of enzymes, of changing the energy of the transition intermediate? Okay. Well, that is a pretty nifty trick. Something that can do 10 to the 17th accelerations is doing something pretty special. How do enzymes succeed at catalyzing reactions? How do they change that free energy of the intermediate? Well, they do it by creating some kind of chemical conditions. And we'll be looking at exactly what kind of chemical conditions in more detail as we look at a specific enzyme. That's true across the board. All enzymes are going to create an environment that's special chemically. Some of them will take cofactors, and so we're going to be running into some of the B vitamins as we go along. Many of the B vitamins are cofactors to enzymes, and they allow for a chemical environment that has a chemistry that just the side chains of amino acids can't account for by themselves. Among all enzymes, we see three mechanisms that are very common. They don't exhaust all of them, but they're very common, so you're going to run into them more than once, and it's worth knowing about them. And that is an acid-base catalysis, covalent catalysis, and metal ion catalysis. And let's take a little look at what some of those things would look like in detail. So for acid and base, you're going to have to depend on amino acids that can either be acids or bases. So you have here aspartate and glutamate in the protonated states. Here's a protonate and histidine. These are all in their protonated states to remind you of their structure. One of the reasons that acids and bases can make that transition intermediate less energetically costly is they can reduce the amount of electrostatic strain on the intermediate. So if we look at the reaction in the absence of a catalyst, here we start off with a substrate of some kind. It's got an R at the end. Could be a lot of different things. Carbonyl oxygen. This is in the keto form here. This is a keto group. To reach the transition state, it's going to have to reach this intermediate where the charge is now sort of spread over it. So there's some negative charge there. There's negative charge there. Negative charges don't like each other, so that's kind of repelling it. And then you get into the enol state over here. And what the acid here has done is to counterbalance some of the negative charge there. Which is going to happen to get rid of this H from the molecule overall. If we look at that reaction now in the presence of an acid, a proton donor, We still have to get negative charges there, but this negative charge now is balanced by positive charge from the proton on the acid catalyst. And so it reduces the strain of this intermediate state. And now the proton that needs to leave can leave, and we end up in the same end state that we reached the other way. It's again the enol form, an ene double bond and an alcohol all. So this is an enol form. So we started with a keto substrate, we ended with an enol product. We started with a keto substrate, acid catalysis, still end with an enol product. No change in the overall energy, no change in the actual beginning and end stuff. The difference is in the exact configuration of the reaction intermediate. This is just showing you how a base can also do that same, can catalyze that same reaction from a keto to an enol. So in this case, you have the base ready to pick up protons. And in that case, you have a situation that might look something like this, where the base can come in at the bottom end, say, and pluck one of the protons off, and in that process facilitate the rearrangement until you end up back with the enol. In all cases, the catalyst is regenerated. That I didn't really play on that. Oops, sorry. Just to sort of point out here, the proton is regenerated for the reaction, so you can go back and do it again. The base can be regenerated. So that's going to be an important part of any enzyme mechanism, is being able to recreate that catalyst. Another very common reaction can proceed through covalent catalysis. And in covalent catalysis, the substrate and the intermediate have to actually have covalent bonds to the side chains of the enzyme. They, for a little bit of time, become part of the molecule. And this is going to look at one particular reaction here, which involves a covalent intermediate in the catalyzed form. Here's a molecule called acetoacetate. If we take a little moment to look at it, it's a 4-carbon thing. Basically, it's 2 acetic acids jammed together. We'll meet this again in metabolism. And the overall reaction that's going to happen here is a decarboxylation, losing a CO2, and ending up with a 3-carbon ketone with the ketone carbonyl oxygen there. In the absence of any kind of catalyst, what's going to happen is a rearrangement where here the electron that's out on the carboxylic acid is coming in to complete that. That electron jumps back there, breaking that bond. That lets go of a carbon dioxide. And then a second rearrangement where the electrons come back in and move on that way. And you can pick up a proton to complete the 3 hydrogens there. So now you've regenerated a methyl group over there. You have a carbonyl oxygen. In the case of a catalyzed reaction, it happens a bit differently. So you come in with the acetoacetate the same. There is a residue on the active site of the enzyme that offers this primary amine to it. And there is transiently an actual covalent bond between the carbon backbone of the acetoacetate and the backbone of the actual enzyme itself. This is called a Schiff base. So the Schiff base then allows further rearrangement. So here's an electron coming in again, popping that back again and up onto the nitrogen. So again, we're going to lose a carbon dioxide. But this is a covalent intermediate here instead of being just a free thing. And another rearrangement can happen with those electrons coming back down and this coming over, and you can end up rearranging again this base until you end up back at the acetone. So I think I've emphasized here the idea that there's actually a bond between the substrate and the the substrate itself, the molecule that's going to be worked on, and the catalyst. In general, there's going to be some electron-rich part of the enzyme that can react with the substrate. S here is substrate. Okay, let's keep that straight. So the most common form of catalysis through enzymes that do covalent catalysis would be a nucleophilic attack. And in that case, there's going to be 2 energy barriers instead of merely 1. So let's take a look at that as we go here. We have the original reactants of whatever they are. You come in and you make your covalent bond and you reach the first intermediate state. And you remember we had a rearrangement of that shift base, so there's a second intermediate in between here. And then that has to rearrange again to get to the other intermediate, which then can relax down into the products. So there are 2 transition energies in the case of something with a covalent catalysis. If we look at amino acid side chains, some amino acid side chains are good candidates for being at the active site of an enzyme that's going to catalyze a covalent reaction. So if we want to go looking for nucleophilic or electron-rich side chains, here are some of them. So serine and threonine are both alcohol groups. This is obviously fairly electron-rich there. And if it picks up another electron pair, it can be negatively charged. Here's cysteine, which is a thiol, which is the sulfur analog of an alcohol. And it also is capable of picking up another electron pair. Here's lysine, which can easily pick up an electron pair that will neutralize the positive charge there when the proton leaves, so you've got that exposed. And histidine also has its 2 states, in one of which it has the electron pair available, either as a pair or as a negative charge, which can pick up a proton. So these are all things that we could imagine might be involved in a covalent catalysis, and when we start looking at some specific examples, we'll see that all of them do at one place or another take that role. Sometimes it's easier to follow this kind of thing if you look at a specific example. And so we're going to look at the enzyme of chymotrypsin in some detail. We're not going to go through this kind of analysis for every enzyme. It is possible to do this for every enzyme that we talk about, but this would be a much longer course in that case. But what I will try to do is, as we go through the enzyme mechanism of chymotrypsin, point out to you features of this mechanism that are general features, things that are true of most or all enzymes, so that you can get your teeth into this particular enzyme mechanism and use it as a sort of a sample of the big universe of enzyme mechanisms. We may look at a few more mechanisms as well, but this is certainly one of the major ones. One of the things that makes chymotrypsin a nice model to look at for enzyme-based catalysis is that it is an example of both acid catalysis and covalent catalysis. So it lets us look at 2 different kinds in the same enzyme. And they were trying to figure out how it worked. And they used a number of reagents that turned out to let them hone in, focus in on specific parts of the molecule. So DIPF is diisopropylphenylfluorine, and you don't need to write it down, just call it DIPF. It's capable of reacting with alcohol groups covalently. And when they treated chymotrypsin with it, only the serine 195 was covalently attacked. So it acts as sort of a pseudo substrate, and it reacted at that site. For that reason, this is called a serine protease. And the serine proteases constitute families of enzymes that are good at breaking down proteins in general. A second reagent that let them start to examine what was going on in this enzyme was something called TPCK. And this mimics a peptide. It's sort of like an itty-bitty fragment of a peptide that's a mimic for it, but it instead becomes kind of a poison to the catalyst and stays attached. And it would react at the site of histidine 57. To finish the general understanding of this catalytic region, they had to go to crystallography. The crystallography has since then been confirmed through molecular genetics, but in a crystal pattern, it was possible to observe a third residue that was part of the reaction, and that was aspartate. And so if we look at this, we have these three side chains of the different amino acid residues. They always have this particular arrangement. And so you have aspartate 102 with one of its oxygens pointing at one of the nitrogens of histidine. And you have the other nitrogen of the histidine pointing at serine 195. So this particular geometry in three-dimensional space was key to understanding how the enzyme could catalyze reactions. And we call this the catalytic triad because of all three of them are essential for this. So let's take a little look at this in detail then. We're going to talk about a scissile bond. So the red bond here between the carbon and the nitrogen is a scissile bond because it can be cut. Scissile is just Latin for can be cut. And in the first reaction of this, what's going to happen is that the histidine is going to pull off the proton from the serine 195, and that makes this a nucleophilic reagent, and the electrons from that oxygen will attack onto the alpha carbon, onto the carbonyl carbon. And that then drives an electron pair off that way. From that situation, we then have a further rearrangement where the electrons that are out there come back in to make that double bond. The electrons in the scissile bond now are the ones that move here and then rearrange like that. So we came in through the carbonyl carbon, we're coming out through the peptide nitrogen. But you do want to notice that at this instant, you have a covalent intermediate, serine 195, that actually covalently attached to the substrate. The next shape it's going to enter into has been called the tetrahedral intermediate. And this is, what I've done is I've copied the transition state from the previous page. It should look the same to you. Oh, sorry. Yeah. So this is a tetrahedral intermediate, and I just copied that over so you could see it on the second page as well. So the name we give this is tetrahedral intermediate because if you were to imagine the carbon atom, it's got, let's see now, 2 bonds out like that, sort of like that. The classic tetrahedral shape of a carbon that has 4 single bonds. This tetrahedral intermediate will rearrange and decompose in the way I was pointing out earlier, that the electrons are going to move in to restore a double bond there. The electrons of the scissile bond will come up and allow the to pull the proton, hydrogen for that nitrogen and restoring the histidine. If you think about it, what that's going to do is this bond is broken now. It doesn't have an attachment to the histidine, so the amino terminus is now free to come off. And it's represented here as a primary amine with just RC. It's the rest of that peptide, however long it was. And so from the tetrahedral intermediate that's covalently attached, we're going to release the N-terminus, the fragment that has an N-terminus just freshly made off into that direction. Now if we look at what's in the active site, it still is covalently attached to the enzyme. And in this case, we call it an acyl enzyme intermediate because this particular configuration, oxygen-carbon double bond there, is the acyl group that we're going to see over and over again. And so this is a fairly stable complex compared to the tetrahedral intermediate because you've gotten the other end of the peptide has moved off and is leaving you free there. And this is the situation that's set up. We don't want to leave it like that because if we left it like that, we would end up with a dead enzyme. That serine would be full and would never be able to do anything again. So obviously there's going to be more happening in the reaction. Somehow or other we have to finish off and release the rest of the peptide from the enzyme. And to do that, we need to take in a water molecule. Oftentimes people forget to actually add in the water needed for hydrolysis, but this one fortunately does. So water molecule can come in, and it's going to be able to pull that electron pair toward the protein, towards the hydrogen. It's not a proton there. Towards that hydrogen, that frees this electron pair to attack the carbon, and that electron pair is driven out onto the oxygen. And you end up with a situation like this, which is, again, tetrahedral. So if you look at that, this carbon has a single bond on each of the 4 possible bonding things. So this is also a tetrahedral intermediate. One of the first intermediates we got to was the first tetrahedral intermediate, which was covalently attached. We're just about to lose the other end of the peptide. Here's a second tetrahedral intermediate. Now if this rearranges, if this electron jumps there, and this electron pair goes into there, what we've done is we've broken this bond right there. And the further rearrangements restore the rest of the active site. And this is showing you that again. This is the tetrahedral intermediate. The N-terminal portion of the peptide is still attached to the enzyme. Excuse me a second. And after those electron rearrangements, now that fragment, that portion of the peptide is free to be released. And what we've done is to completely regenerate the active site of the enzyme. So there's been some parts of this in which protons were attacking, some parts of it where there are actual covalent intermediates. It's an example of needing to use both acid-base catalysis and covalent catalysis. And this is just to break a bond. I mean, keep in mind, all that this enzyme does is break a peptide into 2 pieces. And it takes this much detail to the atoms for the enzyme to pull that off. Well, if the catalytic triad accounts for the actual bond making and breaking that's going on, if the catalytic triad is the place where it's covalently attached, what's the rest of that enzyme for? I mean, if you look at the active site compared to the rest of the enzyme, 3 amino acids out of 80, 120, whatever size a particular enzyme is, there usually are only a few residues that are part of the active site. What do you have the rest of it for? Well, one of the early attempts to try to think about this was the idea of a lock and key, the idea that an enzyme had a special shape to it and that the initial substrates would have a complementary shape the way keys do. This concept was helpful at explaining why an enzyme might attack one substrate and not another. So it was helpful to try to address the problem of specificity. But if the enzyme is fixed in shape, how is it going to fit the products? And do you have the chance of it's actually being too tight? If things fit too well, you have too high an affinity to ever get off of it. And this is an example of what would look like a reaction coordinate under that model, that if you have the substrates bound too tightly to the enzyme, you actually now have a larger transition energy than you had without the enzyme. It would actually slow down the enzyme. So this had some advantages, but it wasn't complete, and it had some disadvantages. One way of thinking about what an enzyme can do is that they can stabilize transition states. So if we're looking here, we're looking again at aspartate, histidine, serine, the catalytic triad. Here's your peptide coming in. Now we're going to pay attention to this little glycine over here. And part of what happens in this tetrahedral intermediate, so this is a tetrahedral intermediate here, is that the hydrogens from these residues can help to stabilize that negative charge through hydrogen bonds. So they're not actually catalyzing the reaction, but they make it more comfy for that tetrahedral intermediate to exist, and in that way lower the energy for it to happen. These are actually called low barrier hydrogen bonds. That is, the hydrogen is equally shared between the oxygen and the nitrogen in these cases. The diagram doesn't quite emphasize that enough. And so this is one way that enzymes can contribute, is they can stabilize transition states and therefore make it easier for them to be achieved. Some of the other things that go on in chymotrypsin specifically, that aspartate 102 forms a hydrogen bond to the histidine that is a bit shorter than a standard one, and this reduces the energy costs for that transfer. If we look at all the enzymes for which we have some understanding, we start to see a number of themes picking up in terms of what can be identified as the things that enzymes do that make it possible for the reaction to go more quickly. And one seems very simple, but it's important, which is just proximity. The closer together reactants are, effectively the higher concentration is, you're going to increase the chance of their reaching their intermediate state. A second contribution from enzymes is orientation, that the way they are in space may be closer to the fit it needs to be. So by holding it in the right position, you're going to make that that much closer to an transition intermediate for it. And those two things alone can account for about 1,000-fold enhancement in many reactions. Just bringing them close together and holding them in the right position. Enzymes do go through other things as well. One thing that's been observed with enzymes is called induced fit. And in induced fit, the enzyme actually changes shape when the substrate is bound. So there may be two or more configurations that the enzyme can be in, depending on the exact sequence of reactions that it's going to catalyze. And in that induced fit, as it's induced to change its shape, it might strain a bond or it might make something more accessible. There are examples of electrostatic catalysis, where the distribution of charges at the active site facilitates the movement of the complementary charges on the substrates. Something that is simple, but you might not think of it, is the active site keeps water out. So when you have our two magical purple and green substrates here in their active site, the packing into that active site is snug enough that water doesn't have access to it. And water can compete for a lot of reactions that might be wanted. The fact that you contribute something by excluding water maybe why, in some cases, you still see the reaction happen, even though you mutate or found mutants that have one of the key elements gone from a catalytic site. So the catalytic site is still contributing proximity, it's still contributing orientation, it's still contributing excluding the water as a side reactant. And I think that's probably a good place to stop for the day. Usually answer questions down here and then I go to my office.